Thank you so much for the introduction. Hello, everyone. My name is Eric Che. I'm a research assistant at the Daniel Bauer and Luca Pinello Labs at Harvard Medical School. Today, I have the pleasure of presenting to you our protocol CRISPR-Clear, in which we attempt to attain a nucleotide resolution screening of the CD19 enhancer using dense base editing and allele-based sequencing. I'd like to begin by introducing our key goal. That is, how can we better understand the function of non-coding sequences? And here, of course, I have depicted a classic model of gene regulation involving an enhancer promoter and several transcription factors. We aim to better understand this model and more specifically understand how enhancer sequence affects binding to transcription factors and how this plays a role in downstream regulation. Of course, we're familiar with common approaches to kind of answering this question, techniques like GWAS, chromatin profiling approaches like ChIP-seq, ATAC-seq, and GROW-seq, as well as chromatin contact assays like Hi-C. These methods can be used to identify functional regions and variants in disease. However, in general, they're larger scale and lower resolution compared to some of the techniques I'll be presenting to you today. So the question is, can we assess regulatory function more directly? Can we tie a more causal, detailed relationship between sequence and function? There are a couple of predominant methods to directly profile enhancers, one being massively parallel reporter assays, where we assess the regulatory activity of several non-coding functions on a reporter gene in parallel. And while MPRIs can be used to assess enhancer-promoter relationships and kind of get a sense of sequence-to-function relationships as well, it's important to remember that it's an ex situ assay, so we can't really get a sense of genomic regulation in its endogenous chromatin context. In order to do so, we can use CRISPR mutagenesis, employing CRISPR to actually edit the endogenous site of interest and observe the phenotype that occurs as a result. Of course, CRISPR mutagenesis can be applied to like a dense screen tiling-based approach in the following way. We can design several single-guide RNAs, mobiling our CRISPR machinery to different areas of a region of interest. After editing has occurred, we typically sort cells based on phenotype, then sequence guide RNAs across these populations, ending up with per-guide enrichment scores across our region of interest. These per-guide enrichment scores are a proxy for the actual editing that's occurring. Because we don't actually observe the endogenous site, of course, quantification of nucleotides and alleles is not possible. Through our approach, CRISPR-Clear, we aim to obtain a higher resolution per allele enrichment score by directly sequencing the perturbed allele, which allows us to perform per nucleotide modeling and ultimately assess transcription factor binding modulation. I'll now begin to kind of describe our approach in detail. CRISPR-Clear is an end-to-end pipeline encompassing both an experimental phase, in which we're generating two different outputs, one using the standard single-guide enrichment sequencing approach, and the other using our more novel direct allele enrichment-based approach. It also encompasses an analytical phase where we've designed several computational tools to process and better understand the outputs we have generated. The experimental approach is a typical CRISPR mutagenesis approach where we're designing single-guide RNAs tiling our region of interest. We transduce this guide library along with the CRISPR machinery into our cells of interest. We sort based on phenotype, usually using fluorescence as a proxy. Then we extract the genomic DNA and divide them half and half to be used in our two separate approaches. There are a couple key differences from kind of a typical CRISPR mutagenesis approach that I would like to briefly mention. Firstly, we're using a base editor, which, as John described, we can use to kind of install single nucleotide edits, either A to G or C to T. We're also employing a specially engineered spry Cas9 that doesn't have the PAM restriction that most Cas9s typically have. And these two changes in tandem allow us to achieve dense tiling of the region of interest. I'll begin by describing the standard count-based approach. So after kind of sorting cells based on phenotype and extracting genomic DNA, we can amplify and then sequence our guides across our populations of interest, ultimately ending up with guide counts across our populations, which we can use to calculate an enrichment score, and ultimately use to get a sense of the editing that's occurring. We can then feed in these guide counts into a tool like CRISPR-Surf, which uses lasso regularization, takes in this genomic tiling screen data, and uses that to deconvolve kind of regions of genomic regulatory activity. It also takes into account the perturbation profile of the CRISPR modality you're using. So in this case, base editings have a relatively narrow region of activity compared to some of the other modalities. And also is able to kind of take advantage of shared data between overlapping single-guide RNAs to come up with an interpretation that, of course, best explains the data. I'll now transition to talking a bit about our allele-based approach. This time, kind of in this pooled screen context, again, we're sorting cells based on phenotype. We extract the genomic DNA, but this time we're amplifying and sequencing the actual perturbed region. And as a result, we can kind of attain a direct sequence-to-function relationship. In a pooled screen setting, this is really exciting because not only can we get per allele kind of counts across populations, we can start to investigate combinatorial effects, as well as hopefully kind of interrogating kind of functional versus bystander effects as well. With this sort of data, the first thing we can do is take these per allele scores and feed them into a pipeline like DESeq2. to get per allele enrichment scores across our populations. And DSeq2 is a computational pipeline typically used in RNA-seq studies to calculate differential expression of genes. It's also been used in CRISPR-based studies and is generally well-suited to analysis of count-based data, so we thought it was a good first approach for this type of analysis. We also have our novel CRISPR millipede-based method where we feed in these per allele scores into our CRISPR millipede method to get per nucleotide level modeling. Millipede is a Bayesian variable selection method. It takes in a matrix of alleles along with allele scores and is able to, through Bayesian regression, regress these different beta scores, where each beta represents a possible base edit, with the score representing how that edit has an effect on gene regulation. Critically, we believe that millipede is potentially able to disambiguate between bystander and functional edits. Of course, given that the data is able to support this type of analysis, because we're proposing this kind of dense tiling-based approach, we believe that this approach is well suited for kind of starting to tease out these kind of combinatorial analyses. Now that I've kind of talked about our method, I'll begin to explain how we've used it to explore the CD19 enhancer. CD19, of course, is a B-cell marker, and clinically it's relevant in CAR T-cell therapies, where CAR T-cell target B-cell malignancies by targeting the CD19 receptor. By analyzing H3K27 acylation data and ATAC-seq data sourced from a study authored by Okayama, we were able to identify a putative regulatory element upstream of the CD19 gene. And then through knockout validation, we observed kind of decreases in mRNA expression as well as MFI, kind of confirming our suspicions that this is likely a putative regulatory element. Now that we've kind of identified this regulatory element, I'll begin to describe our tiling approach. We employed two different types of editors, a PAM-restricted EvoCD editor, and because it's PAM-restricted, of course, we can only obtain sparse CDT tiling over our region of interest. But we also employ this ABE editor with the specially engineered Spry-Cas9 that I described that's not PAM-restricted, which allows us to achieve dense A to G tiling over our region of interest. We use 3 biological replicates, 200 single-guide RNAs tiled over a 315 nucleotide span. We sort by CD19 antibody fluorescence and split the genomic DNA half and half to be used in our 2 separate approaches. I'll begin by talking about our count-based analysis. Again, I would like to orient ourselves by remembering here that the input is kind of guide counts over 2 populations of interest. In this case, a CD19 low population and a CD19 high population. We can visualize these guides tiled over a region of interest and assess their log fold change across the 2 different populations. We can then feed in this tiling screen data to CRISPR-Surf, which is able to deconvolve these genomic regulatory areas of interest, as you can see here. We happen to find kind of consistency between both our AB80 Spry screen and our EvoCDA screen in this region of interest right here, which we wanted to investigate a bit further. So we performed single guide validation using several negative control neutral guides along with a positive control exon 2 targeting guide, and as well as this guide 219, which kind of tiles this region of interest. As you can see from our guide validation, it seems that guide 219 has a significant effect in depleting CD19, similar to that of our exon 2 targeting guide, which potentially suggests that this region is very relevant for CD19 gene regulation. I'll transition now to talk a little bit about our allele-based approach. Again, it's important to orient ourselves and remember that our inputs here are now per allele scores across our populations of interest. We can then feed this into a pipeline like DSeq2 to get p-values and log fold changes, which we can visualize in a volcano plot, establishing some thresholds. We're specifically interested in alleles that cause CD19 depletion. So we can take a look at the edits kind of occurring in these alleles. And we see that most of the edits that seem to be causing depletion happen within this 223 to 230 A to G region. This just so happens to be the same region that we outlined as significant in CRISPR-Surf, kind of showing consistency between our guide-based and allele-based analysis, which is encouraging. We can also take these per allele counts across populations and feed them into CRISPR millipede to get a per nucleotide resolution understanding of this region of interest. And then we can kind of orient ourselves again with the surf results, again highlighting that same region of interest that I've been talking about and zooming in. And briefly now I'll describe how these plots should be kind of understood. On the bottom, we have the sequence, and the vertical axis kind of represents the possible alternate nucleotides that could be installed. So altogether, this matrix kind of represents all possible edits that can be installed in this region. A hit is represented by a colored square, where colored represents effect size. In this case, red indicates CD19 depletion, that this edit is purported to cause CD19 depletion. And then the size of the square represents a PIP score, which reflects our confidence in the hit. Now that we've attained this per nucleotide level resolution, it allows us to do really interesting things, namely motif analysis. So we scanned through PWM motifs sourced from Jasper and CISPP and found that IKZF1 and PAX5 potentially bounded here. We were interested in these motifs not only because their sequence was consistent with the motif, but also because we know that IKZF1 and PAX5 are critical B-cell regulators. We can then kind of compare and contrast our millipede hits with these motifs. I would first like to direct your attention to the AB80 screen. We can see that the A to G edits that have been called the significant hits coincide with the motif. So it kind of makes sense that like perhaps these edits perturb the motif, so these factors cannot bind anymore, which could explain why these edits have been purported to cause CD19 depletion. Interestingly enough, in our EVO-CDA screen, we only see kind of 2 really small hits in this region of interest. And this is kind of consistent, interestingly enough, with the motifs we see here. These 2 hits called here are installing C to Ts, and we see that the motif is pretty allowed to Ts at these specific positions. So potentially, these C to T edits don't, even though they perturb the sequence, they don't potentially affect binding of PAX5 that much, which could explain why millipede has not purported them to be so significant for CD19 depletion. Again, referencing data sourced from the Okayama study, we looked at IKZF1 ChIP-seq data and PAX5 ChIP-seq data, and which suggests that IKZF1 and PAX5 bind over this region that we're tiling. And we also performed some single guide validation. Again, here on the left is our kind of putative motif region. We used a negative control neutral guide, again, the same positive control exon 2 targeting guide, as well as a guide targeting this region of interest, and observed a kind of depletion in CD19, which is encouraging. I'd like to conclude by talking briefly about next steps. We want to perform further guide validation involving IKZF1 and PAX5 cut and run. We want to expand our framework, exploring other readout types and CRISPR modalities, as well as developing an accompanying web application. And finally, we want to apply this framework to other enhancers in blood traits. I'd like to acknowledge the members of the Luca Pinella Lab and Daniel Bauer Lab, specifically my mentor Bashir Bachera, and as well as a special thank you to Sandra from the Davide Ceruggia Lab, who is responsible for performing most of the experiments which I've shown to you today. Thank you so much for your time, and now I can take some questions.
Fantastic. Thank you so much, Eric. Do we have questions from the floor? Microphones are armed and ready.
Hi, this is Chen Yu from UCSF, and it's very interesting results. So I was wondering, for the variants that you identify decreasing enhanced activity and gene expression, do you know if these variants are implicated in any disease?
That's a really good question. That's not something we have specifically investigated for this approach. We are performing other screens which are more I would say designed to target specific variants that we've associated from ClinVar and GWAS studies. But this is more kind of an exploratory approach, and we have not yet attempted to annotate these variants of interest with existing resources, although that's definitely a great idea and something we'd like to pursue in the future.
Other questions? Perhaps I could ask, has anybody thought about doing a head-to-head comparison? There's so many groups now looking at doing MRPA assays that are outside of the cellular context. If you were to do a head-to-head with the approach that you've adopted against one of the more established approaches, has anybody looked, and what differences do you think you might see?
Definitely a really good question. Not for this screen, but again, we're kind of exploring something like that in other screens where we're kind of conducting side-by-side an MPRA as well as this kind of base editing approach. And we hope to kind of tie commonalities between the two approaches and kind of further investigate what's going on, but it's still a work in progress as of right now.
Thank you. Okay. If there are no more questions, thank you so much, Eric. Really appreciate that. And if we could get